Okay. See a decent number of people here, so I'll go ahead and start housekeeping, and then more people can join as they um, get settled as they join, and that way we can maximize the amount of time we have for the actual webinar. But welcome to the Collaborative Science for Estuary series. This is Nick Sobral with the Science Collaborative. Today's webinar features a multi-reserve catalyst project that deployed a novel sensor package designed to measure greenhouse gases across the New England reserves. The team also developed a salt marsh greenhouse gas monitoring protocol for reserves and practitioners to enhance understanding of salt marsh carbon cycling. And you'll hear in more detail from two members of the project team in a moment. But before we get started, I'll give a quick introduction to the reserve system, the Science Collaborative, and this series. So the National Estuarine Research Reserve System is a national network of 30 unique research reserves as shown on this map. This is a NOAA program that works in collaboration with a local place-based partner, either a state agency, university, or nonprofit at each site. Each reserve site includes programs focused on the activities you see on the slide. So that includes land stewardship, research and scientific monitoring, training programs for the public and local officials, and education. The Science Collaborative supports science for estuarine and coastal decision makers by coordinating regular funding opportunities and supporting user-driven collaborative research and transfer activities that address critical coastal management needs identified by the reserves. The Collaborative Science for Estuary series features project teams supported by the Science Collaborative program staff, and partners. Speakers share their unique approaches to addressing current coastal and estuarine management issues. And if that sounds interesting to you, please join us for future webinars to learn about new methods to integrate experts and users into the research process and how their research results and products might inform your work. And we have just so many webinars coming up, so stay tuned for the new year because there's a whole bunch of announcements coming. Before we go any further, just a quick note on informed consent. Webinars are a valuable learning opportunity for our program. Insights from speakers and participants like you can help inform how we support others in doing collaborative science, for example, in the development of future workshops or webinars or resources such as tools, tips, and case studies to add to our guide to collaborative science, which is open access. With that in mind, we want to inform attendees that insights or feedback you share, for example, in chat or via a webinar survey, may be used for program learning and development. Please note that if we capture a direct quote from you that we'd like to use for learning purposes, we will reach out to you ahead of time and seek permission and provide an explanation of its use. All such quotes would be used anonymously. We're grateful for the opportunity to learn alongside you. Quick note on just questions and general housekeeping. All attendees are muted upon entry. We'll be handling questions via the Q&A feature in Zoom. You can enter questions for the presenters as they occur to you throughout the webinar, and we'll try to discuss as many as we can during the final portion of today's webinar. Chat is visible to all, so please feel free to use it amongst yourselves to chat or to alert any of the hosts if you're having technical issues or other kind of questions. And with that, I think I'll introduce our speakers for today. So joining us are Wally Fulwiler, who leads the Coastal Ecology and Biogeochemistry Laboratory at Boston University, and Lena Champlin, a postdoctoral fellow at Boston University, who has helped manage various aspects of this project. And I think Lena's driving, so I will go ahead and stop screen share so Lena can take over.
Thanks, Lena. Thanks, Nick. And thank you all for being here. We are super excited to share with you the work we've been doing over the last year. And I can say that this has been really one of the most rewarding and successful projects that I've been involved with in my career. And I think in large part that's due to the great team that we've had who's been helping us make this project come true. So in that, I just want to highlight, obviously, Lena, who's going to be talking during the second half of this talk with you all. I also want to thank our lead collaborative NERR, which is Narragansett Bay, and in particular, Jennifer West and Kenny Raposa, without whom we would not be here. Obviously, the rest of our NERRs are really important too. We worked with all 5 New England NERRs, and they were just wonderful the entire time and made this whole collaborative project a dream. And I also want to point out and make a special thanks to Dr. Amanda Villard, who has started this journey with me on a previous version of these sensors, and again, without whom we would not be here today. And then, of course, there's always lots of extra support in the field and in the lab. And so science is really best done as a team sport, and this project really highlights how wonderful team science can be. So we have a lot to do today. We have an ambitious hour or so ahead of us. Hopefully we'll talk for less than that. I'll see how fast I can go. So the first part of the presentation, we're going to focus on sort of the research gaps that were identified throughout this project. And I'm going to highlight how the research gaps and the end user needs for the NERRs really coincided really quite well. I'm going to talk a little bit about our iterative process and then pass it over to Lena, who will continue to talk about sort of what we learned from that iterative process and then report some of our preliminary data assessment for what we found. I do want to highlight that this project was 1 year long, and so we really got a lot done in 1 year. And so our data analysis we're just getting started with, and I know you're going to see it and have 1,000 questions, so stay tuned for more on that too. Okay, so starting first with trying to discuss research needs, both research gaps and the NERR needs. Okay, so I think we all are on board with this, but just to make sure that we're on the same sort of starting page, it's important to remember what blue carbon is. And blue carbon is really this term that's used now really to describe basically any type of carbon that's sequestered in a marine or oceanography type setting, right? And the key ecosystems for sequestering blue carbon are our coastal vegetative ecosystems, including salt marshes, mangroves, and seagrasses. The reason they're so good at this is because they're very productive ecosystems. So they're capturing CO2 quite efficiently during photosynthesis. That biomass is being retained in the system, and both the above and below ground biomass contribute to storing and sequestering carbon in the system. Additionally, because of where they are on the border of sort of land and sea, these systems are able to capture particles, both sediment particles and organic particles that have carbon related to them. And so they trap this carbon and store it in their soil. In fact, if you look at where most of the carbon stored in these systems are, it's here in both seagrasses, salt marshes, and mangroves. Of course, today we're focusing on salt marshes, but most of that carbon is trapped in the soil. So you can see that here in this figure that the brown is representing the soil component and the green is representing the biomass component. So they're both productive and they're really good at capturing and storing this carbon. I always like to point this out because I'm in a Department of Earth and Environment that focuses a lot on forests, but I think it's always fun to point out that these blue carbon systems account for less than 1% of the surface area of the ocean, and yet they account for 50% or more of the carbon stored in marine sediments. And so these systems really have a disproportionate impact on carbon storage on our planet. Of course, the thing that makes them really good at carbon storage also sets them up to be areas of greenhouse gas emissions, okay? So you have these areas that have lots of productivity, a lot of carbon in the system, and then that means that there also can be lots of decomposition. And when that's, respiration and decomposition. So the plants are respiring, and then any of the animals in the ecosystem are respiring, as well as the microbes that can degrade that carbon are respiring. And that can lead to a release of carbon dioxide or methane. And we don't really understand the drivers of those releases or the uptake. I mean, we know the photosynthesizing and taking things up, but the rates, as you can see here, for both CO2 uptake and CO2 release are super, super variable. And methane is another component, as is nitrous oxide, but not shown here. We didn't look at nitrous oxide either, that show, these systems that are highly productive are also potentially important sources for these greenhouse gases. And so trying to understand the balance between how much carbon is being sequestered and stored in the system, and then how much is released through respiration is a key goal to understanding sort of the larger blue carbon capacity and climate mitigation capacity of these systems. Okay, there's another issue too that makes things pretty variable. And that is, in our mind, we think like, salt marshes are these beautiful systems, lots of ecosystem services that they're providing, and we want them all to look like this. But in many cases, due to sea level rise, these marshes are drowning in place and we're losing a lot of that productivity. Additionally, and Lena, you can go ahead and do all three of these if you want. Thanks. Additionally, in lots of places, the marshes are being degraded by other human activities, right? So if sea level rise is kind of this large scale regional and global impact, then we have these other more local scale impacts that humans are doing. So in the upper corner here, you can see some marshes in New England, and you can potentially see that there's a road in the background, and then there's a parking lot along the barrier beach. which means that the normal sort of hydrological flow patterns that the marsh would receive, either getting waves and sediment from the ocean or being able to migrate backwards with sea level rise, has been taken away because of the sort of hard infrastructure that we've built. Additionally, below that, you can see sort of this classic picture of what it looks like when marshes are ditched. We did this for a lot of reasons, but a long time ago even, and the scars still remain. So you can see those really straight lines changing how the flow would move through the salt marsh. And then in the bottom here, you can see, we're trying to do things like restore these marshes now. So in this case, this is an opening to allow flow to go back into the marsh. But how that changes CO2 fluxes and what that does, first we have a marsh maybe that's in good shape, and then we have maybe one that's being degraded from some of these human activities, and then we do a major restoration. All of those things are going to be changing the CO2 and methane fluxes, and we don't have a strong handle on how much those things change it. Of course, fixing the flow patterns is not the only restoration activity that we're doing. There are also changes to things like maybe trying to remove invasive species like Phragmites, burning that might be associated with that, or adding different herbicides. And then there's also things like sediment placement, right? So there's lots of these sort of restoration activities that are occurring, and it's going to be important to know how those alter the blue carbon capacity of marshes. So as we started this project, and we talked with our collaborators at all the New England NERRs, there seemed to be these three sort of end user management needs that were the key focus. So there was a strong need for the NERRs to understand and evaluate ecosystem services. And in this case, what we can offer with these sensors are sort of a faster and more efficient way to measure carbon dioxide fluxes. So we can add to understanding blue carbon capacity. There was a strong need to understand how, like I just mentioned, like how these carbon fluxes might be changing with restoration, and how you could maybe measure these carbon fluxes along with also monitoring the restoration, or trying to do it in parallel with other long-term monitoring that takes places at the NERRs, like the long-term vegetation monitoring. And then, of course, there's also interest in trying to understand how climate change, so things like warming, sea level rise, shifts in the growing season, and also shifts in sort of like in more warmer winters, right? We're having like snow, and then it melts and it warms. And so we have like this introduced like pulsing that's occurring in marshes. So there's lots of different climate impacts that are going to change how marshes are functioning in terms of carbon fluxes. And so there's a strong need to sort of understand and document that. Happily, these also coincide with sort of longstanding research questions that we have, sort of on the other side of this. And so, for example, when we're trying to understand and evaluate blue carbon capacity, we're going to understand there's sort of like foundational questions in how carbon fluxes vary across marshes as well as within a marsh. So how much is that variability? Is it more variable to compare marshes, for example, across the New England coast, or are fluxes more variable within one marsh? Answering that is an important question. We may want to do things like, how can we scale these? We would like to be able to measure fluxes all the time everywhere, but that's not possible. And so what are the drivers of these fluxes? And is it possible to use things like long-term monitoring and vegetation or other factors, and then scale the fluxes that we do measure more broadly? And then finally, trying to link some of these climate change questions in, how are CO2 fluxes changing as growing seasons change, as vegetation changes? What do dormant season carbon fluxes look like? These are sort of foundational questions within the carbon community that we have yet to answer. Okay, so let's talk a little bit about the pros and cons of different methods. So first of all, we can all admit that there's like no perfect method, right? And we have to choose methods based on a variety of factors, including access, logistical constraints, financial constraints, expertise, etc. And it would be amazing to have eddy flux towers in all the places, but those are expensive, and they have a footprint that can change, and depending on the different environmental factors. And they're, while they may be able to help us answer questions in terms of like time, so CO2 fluxes over short periods of time in a footprint of a marsh, they don't really help us understand spatial variability within that marsh. And so We might then choose to use another type of method. Next slide. So maybe we would use something like a Picaro or LGR. All of these might be able to get some more spatially refined measurements. However, they're also time-consuming, labor-intensive, expertise-intensive, and they're often cost prohibitive. So while it would be wonderful that for everyone to be able to afford a $40,000, $50,000, $100,000 plus instrument, that's not always the case. So there's a strong need to lower that that monetary barrier to access. Additionally, like we mentioned in previously, there are management needs and questions that are really spatially focused, and we need a spatially discrete measurement in order to answer them. So, for example, if you want to know where to place a restoration site, like adding thin layer placement for sediment or culvert removal or planting or runnels or any of those things, you might want to understand what the carbon fluxes are before and after that. You also might be able to figure out where to do that. Like where is the most efficient, and where do we imagine will have the most success in terms of maintaining the marsh during restoration and also maintaining the biogeochemical benefits of CO2 capture, of carbon capture. And then there are lots of questions about the blue carbon capacity changing over time with something like sea level rise. And having instrumentation that allows measurements more efficiently and easily can enable sort of long-term monitoring or monitoring while we're doing other types of measurements. Additionally, the drivers of carbon fluxes are super complex. And when we sort of imagine what those drivers might be, things like ambient temperature or light levels or pH or salinity, or maybe sediment conditions or plant conditions, we're often measuring those environmental variables when we're measuring a CO2 flux. And that can be problematic. This isn't a table you should dig into or anything, but it's basically highlighting some work a previous PhD student, Holly Emery, did in the lab, where she measured CO2 fluxes really intensely at 4 different sites. And she also measured all of these sort of other variables that we would anticipate be a driver of CO2 fluxes. And despite measuring all of these things, found very low correlations, and nothing really popped out as like being the driver consistently across these marshes for CO2 fluxes. And sort of why would that be? And one of them could be that when you're out there taking a snapshot measurement of carbon dioxide, and you take a snapshot measurement of the light, you're sort of missing maybe some longer term patterns, or you might be missing things like it rained the day before or was cloudy the day before, and there might be legacy impacts of these environmental conditions that you're missing. But one of the many benefits of doing work collaboratively with the NERRs is that there are a host of data, and these are long-term data sets that have been collected that we can link to the CO2 fluxes that we would be measuring. So for example, monitoring vegetation. Vegetation is, we think of it in the lab as like this great integrator of what's happening on a salt marsh over time. So the plants are, sort of always out there, right? Capturing the salinity changes, the light changes, the temperature, sea level, et cetera. And so by linking CO2 fluxes to monitoring vegetation, we think it's a nice way to be able to capture more long-term legacy impacts in the marsh. And then, of course, there's things like monitoring weather at the NERRs, which means we can go ahead and take advantage of site-specific conditions in terms of temperature, precipitation, light, and link those to the CO2 fluxes. So again, one of the many benefits of working with the NERRs are these a really rich data set in which to put these CO2 fluxes into a larger context. All right. Then another major research need, and this links sort of to that, again, to that climate and end user need, is that we really don't have a lot of year-round sampling. So even the eddy covariance towers, the ones that I can think of here, the one that I can think of here in New England salt marsh is not out all year round. That would not be good for the eddy covariance tower because winters can be pretty rough around here. So it would be blown down and lost, which is much too expensive for that. And we've really for a long time just sort of assumed that in the dormant season, there's not going to be a lot of activity. It's interesting that we've thought that because there have been sort of thoughts and ideas that that might not be the case for quite some time. But if you go to the literature and you sort of search for what carbon dioxide fluxes are looking like in the dormant season, there's very few studies, especially in this region, showing any of these fluxes. And so, how can we create something that allows, again, a more efficient and easy access to sampling CO2 fluxes in seasons, which might not be amenable to sampling, but may indeed be important for understanding the blue carbon capacity of salt marshes. Okay, so I want to just start talking here about our iterative processes to design these new sensors, and then I'm going to pass it over to Lena to talk to you more in depth about that. So first, this is sort of the second generation of these sensors that, as I'd mentioned, Amanda Villard was critical in creating the first of these. And now this is like this next set. I also want to really make it very clear that the Octonauts were probably in copyright violation, so they do not endorse these sensors. Although maybe if they ever met us and knew about it, they would. But these are named, each sensor is named after an octonaut because keeping my children interested in science. So what you can see here is our low-cost, ultra-portable sensors. They're small, just about 5 inches by 5 inches. They're lightweight, less than 2 pounds, and they are able to access them quite easily. We calibrate them in the lab, and then we are distributing them to each of our collaborative partners. The variables we set out to measure were time, of course, carbon dioxide, humidity, and air temperature. We tried methane and were not successful this current round, but we are hopeful, and we have a strong lead on doing this, hopefully, for the next round. Okay, so briefly, how this works is we used a typical closed chamber design. So there was a collar that was placed into the sediments, and we used a knife to help sort of open up that salt marsh area. Then there's this clear chamber, and the sensor you can see here, let's tune up in case you're curious. And the sensor here fits inside of the chamber. And then there was a lid that you could attach, and it connected to a phone app, which allowed collection of key sort of metadata along with the measurement of the CO2 fluxes. Okay, and then before I give it to Lena, the process that we used over this 1-year period was to sort of have an initial training, and then we would go out into the field and work with the NERRs, keep people at the NERRs to test the chambers and to test the sensor packages. And then we would have a survey that would follow up, and we would take that information from the survey that asked things like how did you find using the instrument? What would you want to improve? And then we worked on improving those, incorporating that advice. And then we would go back out to the NERRs and work with them with another sort of revised version of the chamber and sensor package. And we did that repeatedly. And so by the end of this project, by the end of the growing season here, we were able to send out the packages to the NERRs and allow them to sort of collect their data on their own. And then once again, we also had feedback from that process. And Lena, I'll let you take it from here.
Great. Hi, everyone. I am going to delve in a little bit more into all those arrows that we saw in that diagram and talk about our process throughout this past year. So one element of our process was having these shared workshops and trainings. So we met virtually on Zoom throughout the year, and this included an initial meeting that had video trainings, and then we also had trainings in the field at each of the 5 reserves in New England. And then our follow-up meetings included reflections on the field work and initial data, as well as thinking about feedback that we received in the field throughout the process and discussing those things. So we met a lot virtually to talk about the project throughout. Another way that we received feedback was through surveys that we spread throughout the year. So we started with an initial survey to think about where we wanted to place our sites based on sampling priorities at the different reserves. And then we surveyed interspersed with field work throughout the year. So we'd have a field trip and then we'd send out a survey that would ask questions about the process of deploying the sensors at the sites and what things could be improved. And then we'd incorporate that feedback and then have another field trip and another survey. And then, as Wally mentioned, that culminated with loaning the sensors to the NERRs and a final feedback survey about that process as well. We also received a lot of feedback about the protocol for deploying the sensors that led to us writing a full written protocol for using the greenhouse gas sensors at the NERRs site specifically. And this protocol includes a lot of different things that we worked on this year, both the deployment of the sensors in the field and how to best streamline that process for having it be easy to deploy the greenhouse gas sensors in the field, as well as collecting associated metadata, which we do with an app called EpiCollect. We also validated these sensor packages throughout the year by comparing them to a backpack Bacaro, and so the written protocol includes information about that process as well. We also developed the process for calibrating the sensors using 3 standard gases and how frequently we should do those calibrations before field trips. The protocol also includes information about how the data is processed after we receive it back from the field and the code that we developed to easily read out the data and calculate the values from it. So our written protocol, we also received feedback from the reserves about that as well. So some of the things that we learned about how many people and how much time is needed to deploy these sensors in the field. And we were loaning out 2 sensor packages, and it's really, we really need like 1 person to run each sensor and chamber. So at least 2 people are needed to deploy 2 sensors in the field. And then there was many different ways that the reserves use these sensors. So you could go out and intersperse these measurements with other field work and just do a few carbon fluxes per day. Or if you're doing just carbon flux measurements, you can do up to about 16 vegetation plots. So that would be 32 carbon flux measurements in one day. And it takes about 16.5 minutes to deploy these 2 sensors simultaneously on average. So as Wally mentioned, at the end of this project, we loaned out these 2 sensor packages to each reserve during their vegetation monitoring times in August and September. And there was a lot of different ways that the different teams incorporated the greenhouse gas measurements into their vegetation monitoring. So some groups had one team that was both simultaneously collecting the carbon measurements as well as doing the vegetation monitoring in the same group of people. Other groups had the same team but different designated roles. So some people would be collecting the measurements from the greenhouse gas sensors and other people would be monitoring vegetation. Other groups had different teams for doing carbon fluxes and other teams doing vegetation monitoring, and they split up those 2 groups. So they were moving at different rates throughout the marsh, and they could move at their own speed separately, but on the same day. And then other groups they did all their vegetation monitoring some days, and then on separate days nearby, they did all of their carbon flux measurements. So just focusing on doing all the carbon fluxes in one day. We also collected data about how easy it is to deploy these sensors. So in our app where we're collecting associated metadata, we had a question at the end of every deployment, which was just rate the ease of deploying the sensors from 1 easy to 5 hard. And so you can see here we got a lot of different scores of just this qualitative measurement of how easy it was to deploy the sensors. And it's fairly easy. The average score was 1.9, but you can see that some sites are harder to deploy the sensors, especially if it's more compacted soil or in the low marsh where there's a lot of mussels. It can make it harder to deploy them, but fairly easy overall, especially compared to other methods. So we incorporated a lot of feedback throughout this year in further developing these sensor packages and the associated protocol for deploying them in the field. And so here you can see both the before and after of the sensor itself, as well as the chamber and collar and associated materials. And I won't go through all of these, but these are some of the major things that we received feedback about and incorporated over time, including having a detachable collar and lid to make it easier to place the chamber and easier to place the sensor in the chamber. And also just the order of things in the field, the steps that we conducted in order to deploy the sensors, we developed throughout this year. And in the bottom, you can see the new sensor design, which has a much more durable design with less wires. And we're working on adding a new and testing a new methane sensor and have the capacity for connecting remotely to this as well. So this is all feedback that was developed through these field experiences with the NERRs this year. Some of the highlights from this project include that during our time period where we loaned the sensors for monitoring, our goal was to have each reserve collect at least 40 flux measurements, which we achieved at all of the reserves, so that was really exciting. We also, in addition to that concentrated time of collecting measurements, we collected samples throughout the year. So we have this year-round data set. We collected samples in December, February, May, July, and then also August and September. And that led to a total of 483 flux measurements, which is incredible considering that there's only a couple of flux towers in New England, and there also prior projects only measure a couple of sites within each individual marsh. It's also really exciting. 26 people helped collect this data, many of which hadn't collected carbon flux measurements before. So it was really exciting to engage more people in being able to collect carbon data. So to summarize all of this and some of the main deliverables from this project, the data set that we collected, as well as that written protocol that I mentioned, will be stored on the CDMO, the Centralized Data Management Office, and available to everyone. We also are creating a short data report that has reserve-specific data and meeting with each of the reserves individually to talk about how the data applies to their specific reserve. We're also creating a video of the protocol that will be shared publicly on YouTube for anyone to access, and are working on publications of this data. So those will be available in the future as well. So with all that in mind, I now want to get into talking about some of our results from this project. So reporting the carbon fluxes that we measured year-round in New England salt marshes. So I just want to jump back to this slide showing our research objectives. And so I'm going to talk through how we addressed each of these different questions, including looking more at the spatial distribution of carbon fluxes within marshes and across regional patterns, as well as linking the carbon fluxes to other factors in the marsh, like vegetation and weather data. And then also think about seasonal patterns of carbon fluxes between the growing season and the dormant season. So as we've mentioned already, we measured at the 5 New England reserves from Connecticut to Maine. So we have this regional gradient in New England, as well as measured multiple sites within salt marshes at these reserves. So we paired our carbon flux measurements with the vegetation monitoring plots in the salt marshes. So these plots are along transects that span from the upland to the primary inundation source. And we collected 2 carbon flux measurements in proximity to each of the vegetation monitoring plots that were in the low vegetation in the marsh, dominated by salt marsh grass and other plant species. And we collected samples throughout the year. So we went to the same sites in December, February, May, July, and then August and September. And then we went to even more sites in August and September for the lone time period, but we went to some sites consistently throughout the year for this year-round data set. So I just want to orient you to, I'm going to be showing a lot of carbon flux data that we calculated. And when we're looking at these graphs, if they're positive carbon flux values, that means that the marsh is a source of carbon, so it's releasing carbon to the atmosphere. And if it's a negative value, it means that the marsh is taking out carbon from the atmosphere. So it's actually a good thing for it to be a negative value because it means it's a sink of carbon, removing carbon from the atmosphere. So here we can see the data that we collected year-round at the same sites throughout the year. And we can see 2 box plots for each of the 5 different reserves. And the 2 plot boxes are one for the summer samples or the growing season samples from May to September, and then the dormant season are in this darker purple color, and that was in December and February. And so here we can see that there's a lot of range of carbon fluxes in each of these sites and a huge span of carbon fluxes even during the growing season for plants. Also, some of these are the initial measurements of dormant season fluxes, and we can see that there's a low source of carbon in a lot of these sites in the winter time, which is really interesting. And especially it's much smaller than the summer uptake of carbon, but it is a significant amount when we look at these relative to each other. So it's very interesting to see these seasonal differences. And just to delve into that a little bit more, we have plots now divided up for each of the 5 different reserves, showing the different carbon fluxes at each of the time points that we measured throughout the year. And these plots show the points, and the dashed line are the median carbon fluxes from that time point, and then the span is the quartile range. And we can see that there's very different seasonal patterns at the site. So along the bottom, some of the more southern sites have a clear uptake of carbon throughout the growing season and a slight release of carbon in the winter. But at our more northern sites, we see some larger seasonal fluxes at Great Bay, where there's a large amount of carbon sequestration in July and a huge seasonal change. But at Wells, another nearby site in Maine, we actually see a more consistent pattern across the seasons and not much seasonal change. And so it's really interesting to see how different marshes that are even close to each other can be in their seasonal patterns. And here, this is really preliminary data, but it's showing the relationship of our carbon flux measurements to some of the meteorological parameters that we have from the weather stations at the NERRs. And so this is just to show that we have this potential to link the carbon fluxes to weather parameters like air temperature on the left and photosynthetically active radiation, or the light levels, on the right. And we see these weak relationships with carbon fluxes when we include our data throughout the whole year. So potentially some of the seasonal differences are being controlled by these changes of weather patterns. But when we look at an individual season, like September, we see that there's a huge span of carbon fluxes that aren't just controlled by the weather conditions. So there's a lot of potential here for us to look more into modeling the impact of various weather parameters on carbon fluxes. So this graph is showing some of the spatial patterns that we see. So just to orient you to it, imagine that you're looking down on each of these 5 different marshes, and along the bottom is the water or the inundation source, and along the top is the upland. And we have along the x-axis our different transects, and then along the y-axis all the plots along the transect. So we measured these paired carbon fluxes by each of the vegetation monitoring plots. So that's why you see the 2 dots next to each other. And just glancing across this, we can see that sometimes even these paired measurements are different from each other. So sometimes they're very similar, but you can have 2 locations very close in space that are pretty different. And there's a lot of variability in space. And you can start to see there's some areas of these marshes that are more of a sink of carbon or more of a source of carbon in red, but that there's a lot of variability and that it doesn't necessarily fall along a sort of traditional marsh gradient from upland to water source. There's really these patchy areas of the marsh that are sequestering more carbon or less carbon. And so it's really interesting to kind of delve more into those spatial patterns. And to look a little bit further into what might be driving some of the spatial patterns, we also looked at how the carbon fluxes relate to specific plant species. So here we can see the different plots that were dominated by different plant species along the x-axis. And on the left, we have alterniflora, which is more common in the low marsh, and then spanning over to the right with patons, which is more common in the high marsh. And we do see differences between the plant species and their relationships with carbon fluxes, and that there was more uptake of carbon in the low marsh, perhaps with more productivity, and then actually a slight release of carbon in the patons plot. So there's a lot of interesting patterns here that we could delve more into understanding why these plant species related to different carbon fluxes. And like Wally mentioned earlier, it's really interesting that we see these relationships because plants are integrating a lot of the changes that we're seeing in the marsh with elevation and salinity and moisture. And so that's perhaps why we see these relationships with carbon fluxes. So then just synthesizing all of that data into a simple mean of carbon flux at each of these reserves, you can see this, these regional patterns of the mean carbon flux at the different sites. And this integrates a lot of different things, including how long the growing seasons were at these sites and when we happen to sample the carbon fluxes here. But we can see that the more southern sites were sequestering a lot of carbon with these negative flux values, and that the more northern sites, perhaps because of their shorter growing season, were on average releasing a small amount of carbon. So now I want to put that in the context of regional gradients. So we can see on the left are 5 sites from north to south, and then again in the middle, this is that mean carbon flux across the sites in these square plots. And there are these regional gradients in New England of factors like tidal range and sea level rise and the area and how that structures the salt marsh. So in the more northern sites in New England, there's a higher tidal range, more macro tidal marsh sites, and also a lower rate of sea level rise. So the rate of sea level rise is actually higher as you get more southern in New England. And then there also is a larger area of alterniflora and more low marsh dominated sites in southern New England. So perhaps these regional patterns relate to some of the regional differences we're seeing of carbon fluxes. And it's really interesting to see, to link the carbon fluxes across these different gradients. So now I just want to end by tying this back to some of the management needs that we talked about at the beginning. So one of them was to evaluate ecosystem services, especially related to blue carbon capacity. Another important need for these New England sites was to think about where to locate restorations and monitoring efforts with blue carbon in mind. And then also to examine how climate change is impacting salt marshes. So thinking about how climate change is impacting blue carbon capacity as well. So just to summarize how our project addressed these needs, we developed this new data set of carbon fluxes throughout the reserves, and it gives a lot of interesting information about how local and regional spatial patterns of carbon fluxes are occurring in these salt marshes. Also, we combined the carbon fluxes with vegetation monitoring efforts in order to be able to link the monitoring efforts with carbon fluxes. And there's also a lot of potential to use these sensors to address restoration needs. So we collected, for example, some before measurements at a site that's going to be restored in Connecticut at Bluff Point. Also, and how this relates to climate change, these values of dormant season fluxes provide a sort of baseline as there's going to be continual change of winter temperatures and winter conditions. So we'll be able to look over time at how carbon fluxes are changing with changing winters. And also the link of these carbon fluxes with vegetation is really interesting because with long-term records of vegetation change, we can see how that might also change carbon fluxes. And with sea level rise, we're seeing changes of plant communities and changes of where the low marsh and high marsh are located. And this has really interesting implications for how carbon fluxes might also change. So with that, I want to just thank our collaborators at the New England NERRs, and then also thank you all for listening today. And if you're interested in learning more about these greenhouse gas sensors or talking more, we have Wally and my contact information, so please reach out to us as well.
Thanks, folks. Great presentation. I saw a few questions rolling in, mostly in the chat. We can keep answering those. I saw that Wally was answering a few in the chat, and I think we had a few in the Q&A as well. But in the meantime, we can move the ones over from the chat at least. So I think I saw a few here. Let's skim through. One person had asked how long they took to put together and if the collars came off. So I think Wally answered that one. Another question was, what vegetation did you cover in the high marsh? I don't know if that was answered or not.
Yes, yeah. So our high marsh plots included some patons, as I mentioned. Some sites have juncus in the high marsh, so we have some juncus as well. There's some disticlus spicata sort of spread throughout the high marsh here as well. I would say those are probably the primary high marsh species that we have. Yeah.
Another person wrote, great way to get these valuable data out. Are there plans to use these for experiments, heat, nutrients, additions, etc.?
That would be awesome. So that would be great. But no current plans for experimental work, but of course, these would be, yeah, a great tool for that.
One person had a general question about the sensors and just wanted to have a little more detail on the sensor itself in terms of how it works. Whether you design them yourselves, or are they commercially available from some company? I think you did answer those in the presentation, but maybe just reiterate it.
Yeah, so we have made those in-house using off-the-shelf components and then tested and calibrated them here. And we are working on trying to figure out the best way to make all of that information available. We do have a paper led by Amanda Villard that's out in Frontiers, and that has a lot of the details in there. And then we're also working on developing ways to sort of streamline the making of these sensor packages. So yeah, please get in touch with us for more details on that.
There is a question in the exit survey that will allow you to add your contact information if you'd like to get in touch about either further questions or potential collaboration. So stay tuned for that. The exit survey will launch automatically when the session ends, by the way, so no need to look for it. Higher tidal range, the more the soil water table should vary. Was soil water table monitored?
No, not by us, not for this.
Yeah, that's a really good potential data source that we can link with. At some of the NERRS sites, there are groundwater wells, so we could potentially monitor the water table at those. But yeah, not all of the New England sites, but that's another interesting source of data.
Another person wanted to know what the approximate cost for the sensors were that were deployed in the chain.
We haven't updated since we've done some changes, but I think between, Lena, you think like 600 or so would be roughly about right?
Yeah, I think the main difference is the addition of the new methane sensor. So I think less than 600 still though, yeah.
Yeah. So if you compare that to some of the other things that are, yeah. 50 to 100K and more. We're doing pretty well.
I was going to ask, what's the comparison for somebody who doesn't know?
Yeah.
Another person wanted to know about your thoughts on trying a giant chamber to capture some of those larger high marsh plants.
Yeah, it's from Molly. Hi, Molly. That would be really fun. So I think our next phase of a proposal that we're currently working on is trying to expand to different sites and to think about how what kind of chambers we would create or could create to do that. So for those of you who aren't familiar with Holly Emery's work, she measured in her PhD Phragmites, and so she had this hugely tall chamber that she measured, so she could capture the whole Phragmites. And theoretically, we could do something really similar here.
Yeah,
I think this is actually a great question. Oh, go ahead. I'm sorry.
Yeah. Oh, I was just going to say, I think in our current chamber design with the sort of detachable lids, there's potential for like stacking the units and covering more taller vegetation.
That'd be cool. There's another question here. I think this is actually a great question because they started it by saying dumb non-scientist question here, and I don't think this is a bad question. Just they wanted to know how applicable this is to other regions, so they're asking specifically about the Southeast.
Yeah, I mean, really, we could use these anywhere you can put a chamber down. We can use these. So Southeast is great. Tropical areas, farmlands, forests. It doesn't really, it's the same idea is applicable in a whole host of other environments, and definitely not a dumb question.
Yeah, and it doesn't even have to be a salt marsh. That's just a place that tends to have low vegetation, but we're using them to measure urban lawns as well in Boston. So applications to other ecosystems as well.
We're working on making a floating one too.
Ooh, that'd be cool too. I see people asking about future collaborations, and I think you said you were, but do you want to say more about that?
Yes, please. Would be the
That's the official line.
That's
Yes,
the
please.
official line. Yes, please.
Yeah, like I said, there'll be a way for you to provide contact information if you're interested in future collaboration. It'll go out in the exit survey, so keep an eye out for that. Got a few minutes left if anybody has any last questions. And of course, you can also ask via the exit survey as well. So no worries. General comment that this is really interesting work. Yes. And this is from Oregon too, so even more. So we've seen interest in the Southeast and interest from the Northwest. So y'all are going to be popular.
Giving me goosebumps. Like the whole dream, right? The whole dream.
Very good stuff. Oh, there's another question. Did you look at dark, so nighttime fluxes at all?
No, we did not. But we talk about that because that would be really interesting. Yeah.
It's a big gap, right? And we talk about whether or not, yeah, we were just talking through that, like, was it yesterday or Monday about it again? We talk about it all the time. So it's a big hole that needs to get filled. And you could do it if you want to be on the marsh at night.
A lot of positive questions coming or comments coming in as well, I see.
Yeah, so Gail, you have a good point, just putting a bag over the chamber or darkening it. But we were talking about that, and it's pretty a different process, right? Because I mean, you can darken the chamber, but it was just exposed to light. It's not really this, you definitely introduce, I mean, you're always introducing artifacts, but you definitely introduce an artifact of being like, oh, it's light time. And then we just cover you up and make you dark, like instantly. And how long the plants take to respond and all of that. But that is the typical way of doing it, right? Wrapping the chamber in something or putting it back.
Okay, we've got a few minutes left, so I think I'm gonna transition us to transition lens.
Good, Molly.
Transition us to wrapping up here. We do have, like I said, more webinars coming up in the new year. So the next one is January 28th. So keep an eye out for those announcements. Other than that, the materials for this presentation will be available in the next few days. Usually just takes us a couple days to get a webinar recording process. You can keep an eye out for the summary email if you register for the webinar. Even if you didn't attend it, you should get the follow-up email from us automatically that has the link to the webinar summary resource page, which will have the recording once it's done. So just keep an eye out for that if you're looking to revisit the presentation or share it with somebody, which it sounds like this is going to be popular and y'all are going to be getting a lot of requests. So it'll be good to be able to point people to this. In the meantime, thank you to our presenters for the great presentation. Thank you all for joining us today, and I hope everyone has a great rest of your day.
Thanks, Nick.
Thanks, folks. Take care.